welcome everybody to day 2 of the Serpentinizing Systems Science Workshop. I was asked to provide a quick recap of what we talked about on day 1. And to do that, I'm going to use notes taken by Katrina Tuing, who's a postdoc in our lab. She is dealing with other things this morning. She'll be joining us later, so I'm going to present a synthesis of the notes that she took. So day 1 was Serpentinization Systems on Earth, so actual field sites on Earth where we are studying serpentinization, and they talked about sites on the seafloor and sites on continents. And each of our invited talks talked about different aspects of those. So first we heard from Susan Lang, who mostly talked about Lost City, an example of a serpentinizing system on the seafloor. She focused on carbon chemistry of what we infer to be a single N-member subsurface fluid supplying the Lost City chimneys. She talks about how inorganic carbon, like carbon dioxide, is very low in the high pH Lost City chimney fluids, which also seems to be a general trend of most serpentinizing systems we've looked at so far. And so instead, organic carbon is much more common than inorganic carbon in these systems, which is kind of upside down from all other ecosystems that basically have ever been studied. She provided evidence based on C13, C14 isotopes of which of these carbon compounds may be produced non-biologically or abiotically, which is important for origin of life and astrobiological purposes. Talks about formate and methane are almost certainly produced by serpentinization reactions abiotically, although that doesn't rule out that methane, for example, could also be produced by organisms to some degree. Some alkanes are probably being produced abiotically, whereas other carbon compounds like acetate and amino acids appear to be mostly produced biologically in Lost City, according to the data that Susan and others have looked at so far. So then we heard from Everett Shock, who used the Samaya ophiolite in Oman as an example of serpentinization on continents. He talked about how the hydrogen concentrations in the wells drilled into the serpentinites in Oman are highly variable, which suggests this biological consumption. So there's ongoing research into that. Everett talked about how calculating potential energy available from chemical reactions using actual chemical measurements of the environmental conditions can constrain which metabolic strategies by life we would expect to be occurring there. He talked about how these systems seem to have a lot of electron donors, so hydrogen, methane, CO, are providing a lot of electrons for metabolic pathways, and so it might be that the availability of electron acceptors like oxygen, nitrate, sulfate might be determining where microbial activity is most prevalent. At least that was my interpretation of Everett's talk, and he can correct me if I didn't get that quite right. And Everett talked about how methanogenesis does not appear to be favorable based on the measurements that have been made. But he talked about maybe that's because methanogenesis already occurred in the subsurface, and therefore it doesn't seem favorable in the places that we actually measure after it's already happened. I see a question in the chat window to ask me to clarify that serpentinization directly does not actually produce methane. That's right. A lot of people like me who are not as geochemically precise use serpentinization as a catch-all word for a lot of reactions that are occurring in serpentinite-like environments. So yeah, serpentinization does not directly create methane, as I'm sure we're going to hear a lot more about that today. So methane is produced by other reactions that are occurring in these environments, maybe not directly by serpentinization. Okay, thank you. I'm sure that will be a topic of discussion today. That was Matt Everett. Then we heard about Matt, who also mostly talked about continental systems, also talked about marine serpentinization systems as well. He talked about how we've only studied a very small percentage of all of the places where serpentinites are found. And there are potentially hundreds more places that we could go around to study, which is an inspiring thought. He talked about how continental systems, one advantage of them is that they have lots of variations in climate, hydrology, chemistry, geology, and use one example of the Santa Elena ophiolite in Costa Rica, where the weather there creates a lot of extreme wet and dry cycles that might give us clues into understanding the hydrology and other aspects of that system. That's an advantage of continental systems rather than the marine systems. He and Everett also highlighted the fact that we have very little evidence of real live organisms that we can grow in the lab from serpentinizing systems. Most of our biological evidence comes from sequencing DNA molecules, which have their pros and cons. One exception is this organism that has been named Serpentinomus, which we call Candidatus because it doesn't may not have an official taxonomic standing quite yet, but it will soon, I'm sure. It's aerobic, so it may not be the best example of the deepest, most extreme anoxic inhabitants of the subsurface, but it appears to be found in every serpentinizing system that I know of. It consumes hydrogen, so it's clearly related to serpentinization in some way. And there was some discussion yesterday about whether it could use solid carbonates as a carbon source, which would be unprecedented as far as I know. Life at high pH is a big challenge, as Matt talked about, and the combination of high pH and very low ions is a double challenge. A lot of the serpentinizing systems on continents have very, very, very low conductivity, low salinity, low ionic strength. They're very, very pure and high pH at the same time, which makes them very different than, for example, alkaline lakes, which have tons and tons of ions as well as being alkaline. So that might be why we see very low biomass in these systems. But these are mostly just speculations. And Matt also touched on some of the many interesting connections to origin of life that serpentinizing systems and organisms have. And then Beth was our fourth invited talk yesterday. She talked about an expedition late 2015, International Ocean Discovery Program, Expedition 357, that went to the Atlantis Massif, which is the giant underwater mountain where Lost City is located. And okay. Where we drilled into the rocks next to Lost City in order to try to get some more subsurface, some direct access to subsurface rocks instead of going through the window and the prism of the Lost City chimneys, which may or may not be the most representative of a more widespread form of serpentinization on a spatial and volume basis. So those rocks were recovered from the seafloor using seabed rock drills instead of a really long drill string that is typically used for ocean drilling, which may or may not have interesting lessons to learn for explorations on other planets. That would be fun to talk about. They did notice already that there were bubbles and water flowing out of the drill holes when they drilled into the serpentinites, which was really exciting. There's a possibility that lots of stuff was going on. And expedition is still in moratorium, still in the 1 year phase of that, although that is ending soon. So hopefully we'll be able to report some results from that expedition soon. And then here I've captured some of the topics that were discussed by the Popup Talks and questions that were asked and discussions that were going on in the chat window as a way of recording and capturing that and encouraging more of that happening today. Katie talked about how different classes of serpentinizing fluids in Oman have distinct microbes. Alberto talked about hydrogen and methane being released from fluid inclusions in deep rocks. Dawn talked about her fresh off the field book results from new serpentinite springs in the Philippines, and Annie talked about trying to cultivate and capture microbes by colonizing them on electrodes in a shallow serpentinite pool in California. And then the other boxes are other topics that were discussed in the chat window. A lot of excitement about the possibility of biological iron oxide reduction, for example, and how we could calculate or explore that. And yeah, I don't need to read all of these. I just wanted to recognize all of the fun stuff that was being discussed yesterday. So I will transition to today by reusing this slide that I used at the beginning of yesterday as a perhaps simplistic way to think about how these sites on Earth could help inform explorations of potential serpentinite sites on other planets, which is this basic intellectual challenge that, for the most part, we are limited by what we can measure on the surface. Even when we drill into rocks, we've basically now created a new surface environment that we are still measuring. In some ways, you can never really get around the fact that you are measuring the surface because we are not subsurface organisms. So that's always the challenge. How do we infer what is really going on in the subsurface and whether there may be some strange green slime down in the subsurface that we can't see? And how can we infer what it is doing by measuring stuff that is leaking out into the surface? That's what we do on the seafloor. That's what we do on land. And hopefully we'll be able to do it on other planets soon. Yeah, I will stop there and make way for all the new exciting stuff for today.
Okay, well, I think I will take over then. For those of you who might not know me, I'm Tom McCollum, research scientist at the University of Colorado, and I'm going to be the host for today's session. Thank you, Billy, for giving us that recap from yesterday. And I just wanted to remind you that these presentations or sessions are going to all be recorded, and I think are going to be made available on the NAI website. So if there's something you saw there that you happened to miss yesterday, I'd encourage you to go there and look at and review that information there. So I'm going to start with giving a bit of an intro to today's session to sort of set the stage for what's going to be happening later on today. So I was asked to organize a session that covered the areas of experiments, models, and things related to that. And of course, just either one of those topics could by itself take a full day of session, a full week of session maybe. So we're really not going to be able to do more than just touch on a few aspects of these topics. And so we have a set of 4 invited speakers and some contributed talks at the end. So I'll just get started and give a little bit of an introduction then. So I think everybody that's tuned in here is probably familiar with this general idea of serpentinization, where you have a mantle peridotite rock that composed of the minerals olivine and pyroxene that reacts with water to become the serpentine suite of minerals, serpentine, some brucite, magnetite, and maybe some other stuff along with that. But as astrobiologists, we're really most interested in these other products shown here: hydrogen, methane, maybe some organics, the kinds of things that can feed biology, could perhaps provide some kind of molecules that could feed into an origin of life. Those are the kinds of things that we're really focused in when we're talking about astrobiology implications of these systems. So I've written here the kind of a chemical reaction, a general chemical reaction to represent this process where we have olivine and pyroxene react with water, become these serpentinite minerals. And again, the key thing that we're looking at in terms of astrobiology is production of hydrogen. The production of hydrogen in these rocks is really closely linked to what happens with the iron, because it's the conversion of an iron, ferrous iron mineral to a ferric iron mineral reacting with water that is what produces the hydrogen. So if you want to think about hydrogen production in these kind of environments in order to feed biology, you really have to link that up with what happens with the iron. The other process that may be going on of interest to astrobiology is the production of methane. And conventionally, people usually think about this as being formed in these systems by reaction of CO2 with hydrogen to produce the methane, although other pathways might be possible. We heard, for instance, at the end of the session yesterday from about Alberto about maybe a different pathway for methane formation. But this reduction of CO2 to hydrogen, this is something that can be done abiotically in these systems, but it also is a potential food source for methanogenic organisms. So really, a lot of the stuff that we're going to be hearing about today, to some extent, is concerned with how you make these things, particularly focusing on hydrogen as a food source, and really what constrains how much hydrogen and where it's produced. So we think about this reaction as being relatively simple. I mean, you're reacting two minerals and producing three products generally, and it seems like it should be rather a simple thing. But it's really not so simple once you get into the details, and I think that'll be reflected in some of these experimental results we see today. And part of the complexity comes in with the fact that when you take iron in its ferrous form out of these minerals, there's a lot of different holes that iron can go to in the products, and they can be in different oxidation states. So you can get iron in serpentine, brucite, talc, magnetite, iwariwite in different oxidation states. And so it can be a rather complicated process to figure out, unravel how that reaction is going to work and how it's going to be affected by different environmental conditions. So just as a kind of reflection of how complicated these things can be when you go and study them in the laboratory, I've shown a plot here from a recent paper by Benjamin Malvoisin that shows the rates of reactions that have been measured, rates of serpentinization that have been measured in laboratory experiments. And I note that these top set of plots up here have a different horizontal axis than the ones down here. And these rates fall into two disparate groups, desperate maybe, but two disparate groups, where one set of experiments measure reaction rates that are really slow, and another set of experiments measure reaction rates that are really fast. So if these ones in the upper left here, these fast reactions are what is really controlling rates of reactions in natural serpentinites, that would imply on geologic timescales that this is a really fast process. Whereas if these are more representative, the ones in the lower right, then serpentinization in geologic systems may be very slow. And there's been a number of studies published since this came out, and again, they fall into these two groups. Sometimes it reports really fast reactions, and sometimes we report really slow reactions. And at this point, I don't think we really have a good grasp of what's controlling whether these rates in the experimental systems are fast or slow, and how that translates into the natural system. Now, again, as astrobiologists, we're really interested in hydrogen generation, and people have been studying serpentinization in the laboratory for several decades, many decades, doing experiments. But most of the older studies were really performed to address other objectives in astrobiology, things like dehydration of serpentinites in subduction zones, things like that. And as a consequence, there was little attention to things that we're interested in as astrobiologists, such as hydrogen generation. So this plot here just shows all the studies that were available as of 2007 that had measured the hydrogen generation during serpentinization. And so this is on the y-axis here. This is hydrogen generation in millimoles per gram of fluids versus elapsed time in hours on the x-axis there. And you can see that at that time there were really just very few studies. And if you could read this legend over here, it would show that all these are conducted at temperatures of 300 and 400 degrees. So if you wanted to know what's going to happen in a near-surface continental system, those things are not going to give you a whole lot of information about what's going on there. So in this next plot, I've shown some more recent results. So over the last decade, people have been more interested in this and starting to measure hydrogen generation rates in different kinds of systems. And the situation is improving somewhat. So you can see now there's a whole range of temperatures covered, different kinds of substrates that have been used in these experiments. And yet there's a lot of disparity, again, in these data. So you can see just like at a single temperature, the hydrogen generation rate from these experiments can range over 4 or more orders of magnitude. And what is causing that variation in these different experimental systems is still under investigation. It's tied up with reaction rates, with where the iron is going, but what really is controlling all of that is still a matter of debate, and especially down in low temperatures where we're interested in kind of continental systems. We want to know if there's hydrogen generated at a rate that would be able to supply these kind of organisms, microbial activities. There's still a lot to be resolved about what the rates are in that kind of environment.
Tom, we have a question from Adrian Brown. He's asking, what does the purple arrow indicate here?
Sorry, the purple arrow indicates that's because that's a maximum value, because hydrogen was actually undetectable in some of these experiments down in this part of the region. The hydrogen generation is so low it's hard to detect, and in this particular experiment here, it could not be detected differentiated from background. So that would be the maximum rate. It's lower, and I should mention these lines are my kind of like simplistic attempt to try and apply first order reaction kinetics and extrapolate from the higher temperatures down into these lower temperature range. And some of these data seem to agree with these extrapolations, and some do not. But these are extrapolations that are really not based on any kind of mechanistic information or anything like that. It's just simply applying first order Arrhenius parameters and doing a calculation with that. So I don't know whether those are realistic or not. So getting into the modeling side, something that has been done with the information that's available out there on the chemical composition of these kind of serpentinizing fluids is to make a determination of the amount of chemical energy that's available to sort microorganisms, just taking kind of a snapshot approach to that. And we saw some similar kinds of calculations done yesterday, presented by Everett Shock from Peter Canovis's work. This is a plot, just shows some similar kinds of calculations done by Jan Amend, published a few years back, looking at the amount of energy available in some ultramafic hosted seafloor systems. But this is something that you can, an exercise you can do to kind of model, to kind of place some constraints on what should be going on there microbiologically and link the chemistry with the biology. And I think we'll see this morning Sanjoy Sam taking these kind of approach a step further to explore kind of the dimension space of metabolic energy availability in these systems. So just to mention the last thing that here that astrobiologists are interested in serpentinization systems is because there are a lot of reasons to think that maybe these kind of systems could have been the locations where the first metabolic process emerged from, where the origin of life maybe got its start. And so we're going to hear some more about that later on this morning later on today from Mike Russell. So these systems that we're looking at today could potentially be a window into the origin of life, both on Earth and on other astrobiology targets. And that's pretty much all I have to say in terms of introduction. And if there are any questions or anything that anybody had to bring up, had questions about that. If not, then we will bring on our first speaker, who is Freda Klein from Woods Hole Oceanographic Institution. If he's there, Freda, are you?
Hello, can you hear me?
Hello, I can hear you.
You can hear me?
Good morning, Freda.
Good morning, hello. And good afternoon to everyone else.
Yeah, I forgot to mention that. Yeah, so good afternoon or evening or whatever your time zone happens to.
So Freda, this is Mike Wyone. I brought your slides up. If you want to start your webcam, that would be great. It should be the button right above Tom's head. Perfect.
All right.
I'm not nearly as handsome on the web as I am in person. But Freda is. Oh wow!
I'm all dressed up.
All right, Freda, it's in your capable hands.
All right. Well, thank you so much. Before I start, I would like to thank the workshop organizers and also acknowledge the contributions of my colleagues and some support from WHOI, NSF, and IODP. So olivine-rich rocks are unstable in the presence of water and at temperatures of about 400 degrees C and below, and undergo a series of dissolution, precipitation, and redox reactions to form serpentinite, a rock that is mainly composed of the mineral serpentine, as we've heard just now. So this process, which we refer to as serpentinization, creates geochemical conditions and hydrogen that make the Lost City hydrothermal system that is pictured here so remarkable. And okay, trying to click on the next slide here.
So, Freda, if you click on the icon in the lower left of your slides, there's a left arrow and a right arrow. Clicking on the right arrow will advance.
Got it. All right, cool. So serpentinization occurs at slow and ultra-slow spreading ridges at oceanic core complexes, and fracture zones, and in tectonic windows at slow spreading or at magma pool passive margins, and in phoric settings of subduction zones. And this map, which some of you have seen yesterday during Matt's talk, shows the occurrence of serpentinites on land. So they occur in Archean and Proterozoic greenstone belts, and in Phanerozoic mountain ranges and ophiolites. And in some of these settings, serpentinization is ongoing, demonstrating that serpentinization is a process that has been active throughout most of Earth's geologic history. And serpentinization affects geophysical properties of peridotite. It decreases the density, the seismic velocity, the shear strength, which can be all nicely shown in the simple box diagram here. And it increases the magnetic susceptibility of the affected rocks, which you can see on the seafloor. So serpentinite-hosted hydrothermal systems like Lost City or Logachev on the MidAtlantic Ridge vent fluids that are strongly enriched in hydrogen and methane and poor in H2S and silica relative to TAC, which is the basalt-hosted hydrothermal system. The pH of serpentinization fluids can range from acidic at high temperatures to alkaline at low temperatures. And to date, the controls on the fluid composition during serpentinization are not completely understood. The hydrogen generated during serpentinization can reduce the oxidized carbon to methane, and if time allows, as Susan has pointed out yesterday, and can also form other hydrocarbons. And the reduced volatiles can support microbial activity, particularly in areas where hydrothermal fluids mix with seawater. And here's an example from the Cretaceous Iberia margin, where we found the fossil remains of microbial communities in serpentinites recovered by the Ocean Drilling Program. The microbes here are trapped in brucite calcite veins that formed at about 65 meters below the seafloor by mixing between oxic seawater and reduced hydrothermal fluid. The biomarkers that we extracted and also measured in situ suggest that these organisms were likely similar to those at Lost City, exploiting chemical gradients to gain metabolic energy. So it turns out that serpentinization does not only support microbes in surface environments, it also does that in the subsurface too. So hypothesis put forward by Mike Russell and others, and I'm sure we'll hear more about that later, suggests that serpentinite-hosted alkaline hydrothermal systems were possible or likely venues for the origin of life on Earth. And in an analogy to Earth, serpentinite-hosted life may also exist on icy moons and other water-bearing planetary bodies in our solar system, and maybe beyond. So taken together, serpentinization is a widespread, ancient, yet still active process with numerous implications for chemical, physical, and biological processes. Yet our understanding of the underlying fluid-rock interactions during serpentinization is pretty limited. So in the remaining time, I will introduce you to some basic petrological concepts and some new results, particularly on iron distribution during serpentinization, the hydrogen generation, and the various pathways of serpentinization. So we've seen similar slides before, actually mainly seen the same serpentinite over and over again, so I thought I would show a different serpentinite here for a change. Serpentinite is the process that describes the reaction between a rock rich in olivine and pyroxene, typically peridotite, with water to form another rock that we call serpentinite, which is mainly composed of serpentine, in addition to a number of other hydrous minerals, oxides, sulfides, and alloys. So in a nutshell, it's a sequence of dissolution, precipitation, and redox reactions, and hydrogen forms as a byproduct of these reaction sequences. So the major reactions can be depicted in the simplified diagram, a ternary chemograph that depicts the compositions of the two primary minerals that we referred to before, olivine and pyroxene, here represented by the magnesium end members, forsterite and encetatite, and the three main secondary minerals in the system, brucite, serpentine, and talc. So if we start with olivine here and add water, we walk across the H2O, towards the H2O apex, and cross the timeline of brucite and serpentine. So that's what we make in reaction one. We can play the same game for pyroxene. So we start with pyroxene, add water, and cross the tie line between serpentine and talc. And that's what's illustrated in reaction two. So only if the right amounts of olivine and pyroxene are reacting, we add water, we will end up with serpentine only. If we want to make talc, we have to either remove magnesium or add silica to the system. So in order to understand the formation of hydrogen, however, we have to consider the distribution and oxidation state of iron and secondary minerals. And Tom has shown this image before. It shows an unhappy olivine that is pitted and that is being altered to serpentine, which is the fuzzy stuff here, and brucite, the tabular mineral, and the octahedral magnetite. All of these minerals contain considerable amount of iron. And to make hydrogen, we need to oxidize the iron originally contained in olivine to ferric iron and magnetite, as shown in the uppermost equation here, or to ferric iron and serpentine, shown in this reaction. So, however, not all of the iron that is incorporated in secondary minerals, here brucite, is being oxidized, and here the example for serpentine. And so that, however, limits the hydrogen generation potential. So this diagram, which is admittedly a little convoluted, illustrates the density magnetic susceptibility relationships of partially to fully serpentinized rocks. And I'll very briefly walk you through it. So we start out with an unaltered peridotite in the lower right corner here, and that is a dense and weakly magnetic rock. And as serpentinization progresses, serpentine forms, which is less dense, and so we move to the left. And if magnetite forms, the magnetic susceptibility increases, and we move up. Right. So, however, in some peridotites that are completely serpentinized, we don't see elevated magnetic susceptibility. Right, it's shown here in the lower left corner where my arrow is. And it turns out that serpentinization is a process that can follow different and quite complex pathways. And the key questions that I want to address is, what are these different pathways and what controls the reaction pathways of serpentinization? Or in other words, what determines which of the many possible dissolution, precipitation, and redox reactions that take place occur? When do they take place? And why is that? So to address this, our approach has to be, or is to integrate results from field observations, laboratory experiments, and theoretical constraints. So thermodynamic reaction path modeling and equilibrium modeling is particularly efficient way to examine some aspects of serpentinization. And without going into the details here, the beauty of thermodynamic modeling is that the composition of fluids, minerals, and rocks can be predicted as a function of, for example, temperature, pressure, and proteus composition. So the way this works for temperature as a key variable, for example, is illustrated in this simple cartoon. We First, we calculate the equilibrium distribution of seawater. Then we add a solid reactant and equilibrate the system. And then we change the temperature and see how the mineralogy, mineral and fluid composition changes as a function of temperature. So in this slide here, in the upper diagrams show the predicted mineral assemblage for serpentinization of olivine here on the left and orthopyroxene on the right as a function of temperature. And olivine is unstable at temperatures below about 320 degrees C and is replaced by serpentine, brucite, and magnetite. So magnetite is this yellow field here. Magnetite is predicted to be stable only at temperatures higher than about 200 degrees C during serpentinization of olivine. During serpentinization of orthopyroxene, which is unstable in this temperature range here, serpentine and talc are the main alteration products, and magnetite does not form at all. So during serpentinization of olivine, magnetite is not part of the predicted mineral assemblage at low temperatures because brucite is taking up a lot of iron and does so increasingly with decreasing temperature. So for comparison, serpentinization of orthopyroxene does not produce magnetite because the iron content or the increased silica activities allow serpentine to contain a lot more iron, which leaves less room or less affinity to form magnetite. So now let's expand this model to CPX, so clinopyroxene bearing peridotite shown here on the left and peroxenite on the right. So serpentinization of CPX bearing peridotite, so Harzbergite in this case here, stabilizes CPX, brucite, serpentine, and magnetite, whereas serpentinization of peroxenite stabilizes talc, tremolite, serpentine, and chloride. The difference here is quite important, actually, for our understanding of serpentinization of fluids, as these mineral assemblages represent strong buffers in terms of pH and silica. So serpentinization of peridotite buffers the pH of the fluids to much higher values and silica to much lower values than serpentinization of peroxenite. So that is quite important for our understanding of the influence of the proteolith composition on the fluid chemistry. So this next diagram illustrates the predicted hydrogen concentration for serpentinization of olivine and peridotite as a function of temperature. Hydrogen concentrations are highest around 300 degrees C at low water-to-rock mass ratios. For comparison, I've also shown measured hydrogen concentrations in vent fluids from seafloor hydrothermal systems, here Logachev, Rainbow, and Lost City. And while the predictions here seem to match the measured values quite well, it is important to realize that changing the water-to-rock ratio in the model allows us to match any measured composition. That means there is no unique solution from the modeling perspective. So in the next few slides, I'll be testing some of the predictions from the modeling in real rocks recovered by the ocean drilling program in a range of different geological settings, here indicated by the numbered circle. So at the MidAtlantic Ridge, at Hess Deep, Iberia Margin, Tyrrhenian Sea, and Mariana Fork. And I'll do that really briefly. So shown here are two backscattered electron images illustrating a magnetite-rich serpentinite on the left from the Mid-Atlantic Ridge, that's the Mid-Atlantic Ridge Cane fracture zone area, and a magnetite-poor serpentinite from the Iberia margin. And while the one on the right does not have any magnetite, it does contain native nickel iron, which is indicative of strongly reducing conditions. So even in the absence of magnetite, you can create strongly reducing conditions. And without going into detail here, so consistent with our predictions, the magnetite-poor serpentinite have actually iron-rich brucite, and the magnetite-rich serpentinite have brucite, iron-poor brucite, if that is clear. And so So that is consistent with a model, but we would like to know whether these magnetite-rich serpentinites that have iron-poor brucite indeed formed at higher temperatures, and vice versa. So we can test that by using oxygen isotope thermometry, and this is a histogram shown for serpentine, seawater, oxygen isotopes from a range of different drill cores. So what you can see is a bimodal distribution with the ones from the mid-ocean ridge setting forming at much higher temperatures than the ones that form at the Iberia margin, where temperatures are much lower and where indeed the magnetite is absent and the the brucite is iron poor. So we can gather a lot of information from the rock record. So we can estimate what temperature they formed, how much hydrogen was generated, and so on and so forth. But some of the aspects are rather difficult to constrain. So what happens, for example, during incipient serpentinization, and what happens over time? Often serpentinites on the seafloor or serpentinized peridiotites are strongly to completely altered, so not allowing us to figure out how we actually start out. So to that end, we conducted several experiments. I'm showing the results of one of them here. So the experiments are conducted in a flexible cell hydrothermal apparatus that allows fluid sampling during the experiment. So this experiment here is where we reacted a solid chunk of Harzbugite with artificial seawater at 300 degrees C and 350 bar for about a year and a half. And we used Harzbugite, unaltered Harzbugite, as the starting material here from the beautiful Merfeld and Mar in the Eifel volcanic field. And the advantage of using a solid rock instead of powder is that the effects of primary textures can be studied, that reaction rates are probably closer to the real thing, to seafloor hydrothermal systems, and that it allows us studying incipient serpentinization. The disadvantage clearly is that the reaction progress is slow and that experiments cannot go to completion on laboratory timescales. So, shown here are the measured hydrogen concentrations as a function of time in comparison to some previous experiments. The hydrogen release is rapid at first, but then tapers off with time, suggesting that serpentinization actually slows down. And for comparison, here are like estimate for reaction rates. So, looking at the measured major element concentrations of the fluid over time, it appears that there's no significant increase in the magnesium or silica over time, while there's an increase in pH, and there's actually a slight decrease in calcium and no significant changes in sodium and potassium. So the speciated fluid composition can be illustrated in an activity diagram here to check out which minerals actually are stable and which ones formed. It turns out that all of the speciated fluid compositions fall into the stability field of serpentine. So when we terminated the experiment, we extracted the altered rock or partially altered rock, I should say, and checked out the surface using an SEM, which is illustrated here in these backscattered electron images. So the alteration minerals include several kinds of serpentine, so lizardite, chrysotile, polyhedral serpentine, as well as calcite, shown here, chlorite, magnetite, and nickel sulfide hazelwoodite. So the other cool thing about using real rocks instead of powders in an experiment is that it allows you to make thin sections. And here we are looking at BSE backscale electron images of the rock interior. And what we see is that the main reaction product is actually lizardite, and which is much more abundant than chrysotile, which forms on the rock exterior. So we find magnetite in contact with olivine. So, for example, here or here, which is in contrast to some of the recent models pushed by Jim Beard, Ron Frost, and others. And we can see that the CPX here is actually stable, so it remained unaltered. And the textures that we see here, bastite textures or mesh texture, are remarkably similar to what we see in seafloor serpentinites. And we find fractures at the end of these cone-shaped edge pits that extend into the rock. Lastly, the serpentinite is only partial, or the peridotite is only partially serpentinized. So there's lots of unreacted material left. So we can approximate the extent of alteration using different techniques such as density, magnetization saturation, hydrogen generation, image analysis, and all of these techniques give us approximately 4 to 5% of reaction. Comparing that with published conversion rates per day, it turns out that the rates that we're getting in the rock chunk serpentinization experiment are really slow. In fact, the slowest measured so far for serpentinization at 300 degrees C. And given that we had no significant changes in the major element compositions regarding magnesium and silica, and that olivine and orthopyroxene are the only reactant, CPX was stable, and serpentine was the only major hydrous alteration mineral, we calculate that this reaction must have led to a volume increase of about 40%, at least under the experimental conditions. There's some discussion about volume changes and mass transfer during serpentinization and in other conditions. So the volume increase led to a decreased access to water to primary minerals and fresh mineral surfaces, also due to filling of fractures and coating effects. And so the system becomes increasingly diffusion controlled. And that can explain the decreased rates of serpentinization on hydrogen release over time. However, we know from seafloor serpentinites that they are actually, in many cases, completely serpentinized. And I think one of the key factors here is fracturing. So fracturing allows water to access the primary minerals and allows these reactions to proceed more rapidly. And that can be due to reaction-driven fracturing, as it is likely the case here, or it can be through tectonic fracturing or even thermal fracturing. So the last point I want to point out, or thing I want to point out, is that we have also some mass balance constraints on the reaction pathways during serpentinization, and this is because we know that the Harzburgite has about 4 times more olivine than orthopyroxene. In order to mass balance this reaction, knowing that serpentine is the only major reaction product, and at least during initial serpentinization, the orthopyroxene actually had to react about 2 times faster than the olivine to make this reaction happen here. But this is only the initial part. What happens next? So during advanced serpentinization, and shown here are the results from a powder experiment using the same starting material, is that that in this experiment, the final equilibrium mineral which was actually reached, which is serpentine, brucite, and CPX, but only after the orthopyroxene was exhausted. So we know the orthopyroxene reacted faster, but it's also less abundant, which means that it will be exhausted first. And then the reaction of olivine becomes dominant. So we have a 2-step reaction sequence illustrated here. And in fact, the the final assemblage here was serpentine, brucite, CPX, magnetite, chloride, and calcite. And the calcite formed under strongly reducing conditions. There was about 100 millimoles of hydrogen per kilogram of fluid. So anyhow, I think it came across that the reaction pathways and hydrogen generation during serpentinization depend on the productless composition and on the temperature-dependent phase relations and relative reaction rates of the primary minerals. Serpentinization, it can be a volume-increasing process, maybe not under all circumstances, in that the rates of serpentinization and hydrogen generation are slower in these rock chunk experiments than in powder experiments. And even though they're slower, they seem to be still fast enough to support microbial ecosystems with energy. And so the rates, I would rather call it actually the reaction progress, seems to be limited by fluid access to primary minerals. Okay, so with that, thank you for your attention and happy to take any questions.
All right, thank you very much, Freda. We do have a couple of questions on the web. One is from Adrian Brown, and he was wondering whether there was any saponite in your reaction products, and if not, why not?
Okay, so saponite is a mineral that we do find in serpentinization systems that have elevated silica activity. And in fact, we did find it in one experiment where we injected a CO2-rich fluid. And so the saponite formed together with dolomite. So there seems to be an effect of silica activity. And the silica activity is influenced or can be influenced by, for example, interaction with gabbroic fluids, but it can also increase due to injection of CO2-rich fluids, due to magmatic degassing, for example.
Okay, we also had a question about what the pH of the fluids in the experiments was.
So the pH of the fluids in the experiments, in the rock chunk experiment, it increased to about 9.5. That's measured at room temperature. However, the powder experiment went all the way up to pH 11 at 25 degrees C, or actually a little higher than that, which is consistent with the CPX, brucite, serpentine buffer. Yeah.
Okay, and one
so
last question.
There's another question about hazelwoodite.
About what?
Hazelwoodite?
Oh, there was. Yeah. Yes, it says hazelwood. I did not know that hazelwoodite is a rare sulfur-poor nickel sulfide mineral found in serpentinized dunite.
Yeah, so the hazelwoodite is cool. So it forms as a reaction product during desulfurization of primary minerals, which can be pyrrhotite, monosulfide solid solution, or pentlanite. And hazelwoodite is one of many possible nickel sulfides that actually do have astrobiological relevance and also to the origin of life, as some of these nickel iron sulfides are also found in the course of enzymes. And
so one last question was about the mineral pairs used in the oxygen isotope thermometry.
So the oxygen isotope thermometry was not done using mineral pairs. It was used. We used Peter Sakosha's 2008 serpentine seawater oxygen isotope thermometer. The serpentine magnetite oxygen isotope thermometer is not experimentally calibrated and can give really unrealistic values if the serpentine and magnetite are not in equilibrium. That's sometimes hard to establish.
Okay, we have time for one last question from Cody Lazar, and he was asking if magnetite-free brucite serpentine assemblage has a wariorite, then what would be controlling the FO2 if not magnetite equilibria?
Right, and so I had this in my original presentation that I wanted to show, but I thought it would get too long. The The controlling equilibria are between iron-bearing brucite and iron and serpentine, as well as, I didn't show this or point this out on the slide, actually andradite in this particular system. So, and we can calculate the hydrogen concentrations that are in equilibrium with the warite and the pentanite, and they, depending on temperature, they were on the orders of tens to maybe hundreds of millimoles of hydrogen per kilogram.
Well, thank you again, Freda. We're going to move on now, and our next presenter is going to be Lisa Mayhew here at the University of Colorado. So I will turn it over to Lisa.
All right. Thank you very much, Tom. I want to thank Tom for inviting me to speak here, and also comment as I figure out how to make my slides.
So the arrows there.
That I'm a research scientist in Alexis Templeton's lab here at the University of Colorado in Boulder, and I'm going to be talking about work that I have done in close collaboration with Eric Ellison and Hannah Miller, who are both members of Alexis's lab as well. And we've each sort of spearheaded different parts of the work that will be presented today. So I know these things.
You can use the keyboard, the right arrow from now on.
Okay, great, thanks. All right, so Freda made my job of introducing this so much easier than it would have been had he not gone first. But just to remind everybody that if we take a look via sort of I think this is 10 or 20x photo micrograph in plain polarized light of a partially serpentinized rock, we can see that they possess a complex mineralogy. And we're hopeful that this complex mineralogy is representative of a reaction path history that they've undergone in a way that we can actually unravel through use of a number of different analytical techniques to sort of understand the pathways that these rocks take to go from an unaltered peridotite to an altered serpentinite. And we need to figure out how the primary phases like olivine and pyroxene and chromite are related to secondary phases, for instance, serpentine and magnetite. And my main interest is trying to tease apart the redox transformations of iron that take place during these reactions at low temperature. And I'm not going to go into these plots in great detail because Freda did, and they're his plots. Where's my pointer? Okay. But again, you can see that magnetite, in terms of being a phase that precipitates out during serpentinization and that can accommodate iron 3 that forms during the reaction or from the oxidation of iron 2, really doesn't become much of a player until about 175 degrees C. Whereas brucite and serpentine are much more prevalent at these lower temperatures. And then in this plot, Freda has shown essentially the contribution of iron, or the amount of iron in these different phases. And you can see that brucite contains iron at much higher concentrations at lower temperatures. And these are the temperatures that I'm interested in thinking about these serpentinization reactions. And so a question is, at these low temperatures, what phases are the reservoirs for iron 2 that might be reacting, especially if you're starting with an already partially reacted rock? And then what phases form and can accommodate iron 3 as that reaction progresses? And so the challenge, and then if we consider other possible alternative secondary phases that might form besides magnetite, which is shown in black. This work by Wolfgang Bach recently was kinetic models, whereas Freda's models were thermodynamic equilibrium models. You can see that we can make hydrogen if we put iron 3 into phases other than magnetite, including things like iron 3 oxides like goethite and hematite, and also kronstatite and iron end member, the iron 3 end member of serpentine. So how do we unravel all of the relationships of the mineralogy, and how do we do it on a micro scale? These are the questions that we're trying to work on at this point. And so a key component of this then is having the appropriate rock samples to work on. And yesterday, a number of the systems that we're studying were introduced by folks like Billy and Beth and Everett. And so I'm just going to talk briefly here about Lost City. And again, Billy mentioned that really most of the previous work has been done on these chimney structures. And these are images from the cruise to Lost City back in 2008 that I went on. And most of the work then focused on getting fluids. Oh, that didn't. Sorry, it's not quite the right order, but fluids from these chimneys. And I was interested in actually obtaining the underlying rocks because I wanted to know, even back then, what do those rocks look like and how might they be producing hydrogen. But at that time, all we had access to was taking the ROV Jason and trying to get him to pull rocks out of the fault scarp that underlied Lost City. Whereas with the expedition that Beth spoke about yesterday, of which I'm a science party member, we were actually able to drill down into the subsurface of Lost City and get these drill core materials that now we can study in more detail. And we can see, even from a plane polarized light photomicrograph, that they're very complex materials. And so we're really excited to have these rocks in hand now. And they're some of, I think, the most promising and exciting samples we have, especially because the work that I'll be doing on iron speciation will be coupled with work such as Billy and Matt are doing with molecular, and Beth, molecular and microbiological techniques to try to correlate with what organisms might be living in these systems, and also work with Benedict Menez from IPGP in Paris to look at the carbon speciation. So there's a lot of, I think, power in these sorts of expeditions where we can all work on the same samples. But all that being said, I'm not going to talk about those today at all because they're still a work in progress. So then moving to Oman, Everett introduced this system to us yesterday with these beautiful images of hyperalkaline springs seeping from the subsurface, and mentioned also showed plots of the hydrogen that they measure from in these sorts of waters and samples. And again, the question here that I'm interested in is, what are the processes that are going on in the subsurface that produce that hydrogen? So the Oman drilling project, which just got started in December of 2016, is now pulling up some of its first few hundred meters of core. This is actually a photo of Olivine Gabros. And I think you heard a little bit about this from Katie perhaps yesterday too, but we're going to have access to core from actively serpentinizing regions in Oman. And we're really excited, again, to look at these rocks in the context of also having coupled geochemical and microbiological measurements on them. We're excited about this because then we'll be able to investigate the mineralogy and able to try to pin down what processes are making the hydrogen that then Everett and others are measuring in the springs and in the well waters that are being sampled. So this does beg another question, though, and that's what mineral phases, again, actually are preserved in these rocks that might represent the reaction history. It might be signatures of what's happening that are representative of past hydrogen production. And so just to tie it back to astrobiology, it's these sorts of questions that we hope to inform when we think about looking at or for these minerals on other planets. Beth and Yelman's work has, I can't tell what my pointer is, has done some remote sensing and detected serpentine minerals on the surface of Mars. And so what does that detection represent in terms of a reaction history? And does it represent past hydrogen production, perhaps in the subsurface of Mars? And we want to inform that discussion as well. Okay, so we haven't had access into much of those subsurface materials until really recently. So we've been developing our analytical techniques and conducting our experiments on the wide range of surface materials that we have available to us. And in that case, Oman is a great case study, sort of natural laboratory, where we see a wide diversity of rock types and the chemical complexity of serpentinites. And you can even see that at the macro scale here. This is a relatively unaltered peridotite, and this is a much more highly or extensively altered peridotite. And so we're going to start by looking at these rocks at the bulk scale with some techniques that can shed light on the redox state of iron in those rocks. And so we mainly use synchrotron radiation-based techniques to get at mineral speciation coupled to the iron oxidation state. And I'm going to go through some of those techniques here. Using these fluorescence spectra, we can actually see a difference between, in green here, we have olivine, and in red is ferrihydrite, so an iron 2 bearing and an iron 3 bearing phase, with blue in the middle here being serpentine. And you can see that the peak position changes, moves from lower to higher energy, depending on the oxidation state of iron. But this is also complicated by the coordination state of iron and the mineral species that the iron is being held in. And so if we want to get, well, but still we can get some information from this. And when we collect these full spectra, you can see that these less altered rocks kind of group over here in these red, orange, or I'm sorry, black and orange spectra. And these more altered rocks make a group, again, at higher energy in these lighter color spectra. So we can definitely tease apart differences. But we can turn to the pre-edge down here of the spectra, which is blown up over here, to be more quantitative about, to be more quantitative about the difference between the redox state in these different mineral phases. And so this is just a blow up, essentially showing you the difference in the pre-edge region of the structure. And the pre-edge is more sensitive to the oxidation state and the coordination in a way that's actually predictable and can be quantifiable. So, briefly, what we do is collect these Zane's spectra on our bulk. These are bulk, thank you, samples, powdered samples of those rocks. And then we background correct that part of the spectrum, and we figure out what the centroid of that peak is and the integrated intensity under that peak. And we can plot that onto a variogram. And here we're really building off of the work done previously by Max Wilkie and Muriel Andriani, and using standards that were provided to us by Manuel Munoz to quantify the iron 3 in our samples. And so we use these standards that are N members of iron 2 in octahedral coordination or tetrahedral coordination, iron 3 in octahedral coordination or tetrahedral coordination, and then these manual mixtures of those standards then that give you a quantifiable sort of estimate, I guess, of the percent of iron 3 in your unknown sample. Okay, so when we do this with our bulk Oman samples, we find two separate populations, which is good because that's what we're seeing from our other techniques as well. And that's these less altered peridotite tend to have 30 to 50% iron 3, whereas the more highly altered peridotites that we looked at have greater than 80% iron 3. So there's a definite difference in the oxidation state of iron in these rocks on the bulk scale. Okay, but we want to know where does this iron actually reside? Which phases are actually have the iron 2 and which ones have the iron 3? That's interesting. And so this is a ChemScan map, microscale map of mineralogy in one of our less altered peridotites from Oman. And the green is olivine and the brown is serpentine. And so you can see they're already extensively serpentinized, but there's also a lot of relict olivine around. And that looks very different from one of these much more highly altered rocks. And this is an example of one that's extensively carbonated. That's in purple, all of the carbonate. Has about the same amount of serpentine but no olivine, and the pyroxenes are more highly reacted. So there's definite differences, significant differences in the mineralogy of these two different rock types from Oman. And then we've turned to hyperspectral Raman imaging to try to get at the mineralogy in even more detail. And this has revealed things to us that we couldn't see from ChemScan analyses. For instance, we find minor phases that are interesting for a lot of reasons, that including possible carbon associations such as garnet. But what I want you guys to focus on is the serpentine, and there's two different serpentines, one that has this extra peak and one that does not. And you can see that they're intermixed in this cyan and the dark blue here in this Raman mineralogy map, which is what's shown here and corresponds to this color legend. Right, we can then look, and I guess I should back up and just say that this map and these spectra were collected within the fingerprint region, which is a pretty diagnostic region of a Raman spectrum for distinguishing between different mineral types. But we wanted to know more about the difference between these two serpentine phases. And so we also mapped out at where the OH bonds are active, and we actually see a whole different peak that is much more intense in the areas where serpentine is blue. And this corresponds actually to the position of the OH peak in brucite. So using this Raman technique, we're able to distinguish that our serpentine in red is different from our serpentine in blue, and that one key difference seems to be the presence of brucite mixed in with that serpentine. And happily, Freda explained a lot about the potential importance of brucite because it can take up iron too. And Eric, who is in our lab and has been working really hard on coming up with a method for actually being able to estimate the iron content of this brucite, even when we can't take these samples to the microprobe and make individual chemical measurements of these individual phases because they're just too intermixed on a micro scale. We can use the position of that peak in the OH stretch region of the Raman spectrum. It shifts in a predictable way with iron content. And so we're actually Eric was able to build a calibration curve of the position of that OH peak relative to the magnesium number of the brucite that's intermixed in that serpentine that we see. So this has given us a window into understanding how much iron might actually be in these brucites that we're finding, again, even though they're really closely and intimately intermixed with serpentine. Okay. But again, we want to know more about the redox state of the iron in both the serpentine and the brucite. And so what we've done is then adapt that pre-edge technique I was telling you about earlier, which we did on these bulk spectra that we collected for bulk materials. We want to adapt that for our map areas. So going back to this map here, can we actually figure out the distribution of iron 3 within these different mineral phases and their locations? And so what we do here, we don't have access to a hyperspectral mapping beamline, or not easy access anyways. So what we've done is take smaller map, or not smaller maps, but take a single map, okay, at a number of different energies throughout the pre-edge, and then sort of build or fit a peak to those energies. And the same process that we did before, we background correct. We can then calculate the centroid and the integrated intensity and plot this data on a variogram. But now, instead of just a couple data points on a variogram, we actually have this whole cloud of data where each point corresponds to a pixel in our map area. So you can see the spread. Oh, sorry, thanks. The spread in iron redox state represented by the different phases in this map. And we can calculate then the iron 3 over total iron ratio for every pixel in the map. And transpose that so that we can map out the redox state of iron onto the map that essentially that map that we collected. And this is even more information because it takes into account not just the iron 3%, but also the iron concentration overall. And so the way you would look at this is olivine here has more iron 2 in it, and serpentine has more iron 3 in it, and these sort of chromites and things have less iron in general. So we have learned a lot more now about what the iron oxidation state in our different mineral phases is and how it's distributed. And you can see there's variation. It's heterogeneous within the serpentine itself. Okay, so Mike, if you could start the movie. What I'm going to try to explain to you here, and I should probably just make Derek do this, but is what we're doing in this movie is actually overlaying a redox map that the redox map that I just showed you with the mineralogy map, because there's just a lot of power in being able to say, okay, I don't know if I can click or not.
I don't think. Oh yeah, you can do a pointer.
Oh, I can. Okay. So these, if you look at these red stripes here, that's the serpentine, and it appears to line up with areas that are more oxidized. And the blue serpentine appears to line up with areas that are more reduced. Okay, I think we can go back.
Cool. I didn't know you could do that.
Yeah. Okay, so serpentine, the red serpentine is more oxidized than the blue serpentine. And this makes sense. The blue serpentine has that brucite in it, which is iron 2 bearing. And so another way that we can represent this data quantifiably is by plotting the density of pixels for each mineral type. So we're actually now comparing olivine, the iron 3 content of olivine to the different types of serpentine that we see. And we can see there's definitely a shift in that peak position. So anyways, I know that was a lot of information really fast, but I want to get on in the last couple minutes to some other work that we're doing. This is that same rock that we've been studying in very, very great detail. And Hannah Miller in our lab has been working to react that rock with water to actually see if it can make hydrogen. We know there's brucite in it. It's iron 2 bearing brucite. Can we crush this rock up? I mean, it still has olivine in it as well. And can we make hydrogen at relatively low temperatures? And we do see significant and sustained hydrogen production at 100 degrees C. These are triplicate experiments with different media, a seawater media in blue and a rainwater media in green, with hydrogen plotted on the y-axis here. And we see sustained production of hydrogen over a number of months. So that's really cool. And so we're trying to then, can we interpret what reactions are happening that are creating that, leading to the production of that hydrogen? And here we can just look at some relatively simple XRD spectra and see that this brucite peak that exists here in the unreacted material is actually missing, disappeared from these reacted experiments. So it seems like brucite is consumed during these water rock reactions, which is pretty cool. And I'm not going to show you the evidence, but there's serpentine and magnetite that form, and we'll move on here. But one of the big complicating factors in these sorts of experiments is the experimental setup. What I'm showing you now in this plot is the concentration of silica increasing with time during reaction in these glass serum vials in which we're doing these reactions. We've also documented hydrogen input and loss across the rubber stoppers that cap these vials. So this is like the easiest way to do these experiments is in these glass vials, but there's a lot of complications involved. So a while ago, we started trying to figure out how to address those. And this has been a work in progress for quite some time. But we are developing a titanium reaction vessel that does not have any rubber or anything like that, but which also allows us to sample repeatedly for gas concentration without contaminating the system or leaking any gas or adding any gases, and then also will allow us to get the fluids and the minerals out of this system as well. And so this is a work in progress. Okay, so anyways, sorry to be too long, but this is just a picture of what we all have going on in the lab to push forward work both analytically and experimentally to try to get at these low temperature water rock reactions.
Okay, thank you a lot, Lisa, for that presentation. And we are scheduled to take a break now. Were there any questions that came up? That
we have 3. Wouldn't mind full screening there.
Perhaps be answered. So what time? The time is now 11:20. We will reconvene then at 11:30, and I will let Lisa answer the questions that came up if she wants, and those of you who want to take a break can go ahead and do so, and Lisa can address the questions, and if you could read the questions. And
yep. I'll read them for you if you want.
Sure.
So the first is from Adrian Brown. He asks, did you find non-ternite in any of your samples?
So Adrian, if you're referring to the serpentinites from Oman, no, we did not. If you're talking about experimental samples, we sometimes do find clays because we're adding extra silica into the system from the borosilicate glass vials.
Okay, and this is a question from Freda Klein. He asks, Lisa, how do your Zane's results, Xanes, compare with Muriel's results? And did you find any iowate in your samples? Iowate. Thank you.
I guess, Freda, that question of how our Zane's results compare with Muriel's, I'm not quite sure how specific you want me to be. We're using a bit of a different approach when it comes to the mapping because they have access to hyperspectral mapping results, and we're studying different sample suites. But we've presented this data at Serpentine Days and had long conversations with Muriel and Manuel Munoz, and I guess are sort of moving forward in understanding how and where and how we have discrepancies in data sets that maybe don't make sense, and we'd like to actually cross-compare between data sets that they've analyzed in their way versus ours. And Freda says, in terms of overall iron 3, iron total in different rock domains. Not entirely.
Serpentines fell in similar fields.
Yeah. So the serpentines seem to fall in similar fields in general. Okay. And then Iowaite. Do you want me to?
I'll just say that yes, we do find interesting indications of things like iowate or other hydrotalcite group minerals. Not in particular in the sample that Lisa was talking about specifically, but we do find those sorts of minerals in other rocks from Oman.
Okay, and the last question is from Hector La Madrid. Going back to the Serpentine 1 and 2, what is the paragenesis? Serp 2 was the more oxidized?
No. Now I just need to think about the legend. Those are unfortunately named. Serpentine 2, I believe, was the serpentine plus brucite, which is the less oxidized. And in terms of the paragenesis, yeah. I mean, like the different generations of serpentine, is one the first versus the second generation of serpentine? Because I'm not quite sure what you're asking. I know, know, that's why I can't.
We'll let Hector clarify his question.
We're laughing because her son is typing in. It's her 2-year-old son is typing in a question.
She's sponsor.
Yes, SERP1 being first, right? Okay, so unfortunately the numbers of SERP1 and SERP2 don't correspond to the paragenesis or the generations of those serpentines, and I actually can't speak to that right now. I guess I'm sorry about that. Okay. So Brian said
we might have
to.
Just so we can get people to have a break. Do you want to maybe take these in the chat and we'll
Sure.
have a quick 10-minute break?
Good.
Yeah.
Okay. So we'll
Thank
see
you,
you all.
everybody.
Let's all thank Lisa one more time. So it's now 10:25 Pacific. We'll be back at 10:35 Pacific time.
So thank you very much for having me, and thank you, Tom, for inviting me to be a speaker here. I'm very grateful also for Tori and Jen to organize this workshop, and a big shout out to Alexis for organizing this cohort of serpentine enthusiasts. So if you don't know me, my name is Sanjay. I'm a research scientist with BMSIS, and I'm working at NASA Ames and Tori's group. And the title of my talk is Biological Potential of Serpentinizing Systems. It's a title that it's quite similar to Everett's talk yesterday, and for good reason. We use complementary approaches to kind of understand the big question of how to link microbiology and geochemistry. So how does one actually do that? Everett kind of gave the answer yesterday, and that is through energy. So rocks and water and heat are the source of energy then that biology consumes. So you can describe this water-rock reaction in terms of joules, and you can describe the biology's use of what the water and the rock puts out also in terms of joules. And so energy is linked quite a bit to habitability, which is why we're interested in this in the first place. And so the next question is then, how does one quantify this concept of habitability? And different people have quantified it differently. Carol Stoker has a definition. Dirk Schulze-McCush has another one. Everett also has another one through the Shock and Holland 2007 paper. And Tori, through his 2004-2007 papers, also has his take on it. And of course, being a little bit biased towards the latter one, is what I'm going to be talking about. I'll give the answer and how does one actually quantify this concept of habitability in 2 slides. But first, I want to define a few. Ooh, already? I think there's a slide missing there. But what I want to do is, okay, so let's just wing it. And in order to first understand how this quantification is done, I want you to kind of take the analogy of an electrical circuit and apply it to a biological cell. So you'll see in the parenthesis under my cell, I've just said a cell. It's ambiguous for a reason. It could be a photovoltaic cell. It could be a biological cell, because the analogy is quite good. So on the left column, you have the properties of an electrical circuit, and on the right column is the corresponding properties of a biological cell. So they both use energy, right? Solar, geothermal, add nuclear to that if you wanted. If you're talking about an electrical circuit in particular, the use of that energy is, in this case, light. On the biological side is biosynthesis, of course. And you can describe this cell using electrical properties such as voltage as well as current. And the analogy in the biological world is that gives free energy. So that's the affinity that Everett was talking about yesterday. So think of it as a chemical voltage. And the flux of electron, the current, is the flux of substrate, which are carrying the electrons into the cell. Of course, redox reactions are essentially electron transferring reactions. So the analogy holds there as well. So here are the keywords that I wanted to make sure we are on the same page, because I'm going to be using them quite a bit. The first one is the concept of maintenance energy. So it's defined as the energy required by biology to maintain status quo, as in no growth. In other words, it's the energy needed by biology to fix what the environment breaks. So you can kind of see that it's related to temperature. The higher temperature of the environment, the more work a cell will have to do to keep the house in order. The next concept, of course, is the Gibbs free energy. Again, it's what Everett yesterday called affinity. It's the energy made available by the environment that can be harnessed by biology to do work. And that's important because the environment supplies a bunch of energy, much of which cannot be harnessed by biology. Think of the heat, for example, the geothermal heat. So one way to frame it in your mind with the analogy of the electrical circuit is you can think of Gibbs free energy as the chemical voltage, if you will, of a system. And I define here a concept that Tori has defined as the habitability index. It's a ratio of the energy consumed by biology over that maintenance. So if that ratio is greater than 1, then we can define the environment as habitable. If that ratio equals 1, you're essentially getting from the environment as much as energy as you need to maintain yourself. And so you're habitable, but barely. And below 1, energetically, you're considered not habitability. So that's kind of the framework that will work around. And so the methanogenesis provides this great laboratory to study this link between biology and geology through and focusing on methanogenesis, because as we've learned this week, serpentinization produces hydrogen, and methanogenesis consumes this hydrogen. So you can make that link between geology and biology through hydrogen. So what has been done in the past to try and quantify the energetical potential of springs is calculate that Gibbs free energy. So on each row is a different spring here, and the first 3 are the velocity hydrothermal vents, and on each column is a different property of that spring. So you have hydrogen, pH, DIC, and so on, and you can calculate delta G, right, a chemical voltage, if you will, of those springs, and then look at the different metabolisms like Everett talked about yesterday and see which ones are energetically favorable. But you can take a bigger picture. Take this table and put it in a larger scope. So what I've done here is taken hydrogen on the y-axis, which is flat in this case, and pH on the x-axis, and plotted that delta G that gives you energy on the z-axis. I didn't necessarily have to do it in 3D, but I really enjoyed plotting. Don't judge me, and so this is why it's like this. And so one thing to remember about delta G is that it's the negative delta G that we are interested in. So that's the good voltage, right? So anything that's higher than -10 is roughly it's the kind of the minimum threshold to run ATP. So the voltage you need to apply to run ATP. And so anything that's higher than that, that's kind of the yellowish red in this plot is death, you can see. If I kind of roughly show the location of the physicochemical space of lost city, you're in here, and you can see that delta G with respect to pH doesn't change very much. And it's kind of moderately affected by change of hydrogen. So delta G is a great way to kind of scan the landscape quickly, but doesn't really give you a kind of a high resolution to understand whether or not an environment is really habitable for a particular metabolism, because it doesn't take into account the energy needed to survive in that environment in the first place. So that's where this habitability index comes in handy. So so if you look at the again, returning to our analogy here, we've been focusing on the last two, the electrical properties. The voltage, again, is that Gibbs free energy that we can be easily calculated. And but if you measure just the voltage in an electrical circuit, you could tell that there was something there, like a light bulb, but you wouldn't be able to tell if it's a 60-watt light bulb or 100-watt light bulb, 120-watt light bulb. But if you were able to measure the current, then the product of the current and the voltage is the power. And so then you can be a lot more specific about what's going on. And so how do you then calculate this current, this flux of substrate in a biological system? Well, you can imagine if this biological cell this time was sitting in and was eating it, there would be a gradient in yumminess between the exterior and the interior of the cell, right? It's a highly technical term. I hope you're following. And so this gradient, you can quantify it mathematically as the diffusion of the substrate from the outside to the inside. And interested in the details of this, I'll refer you to the magnus opus of Alperin and Holler 2009. But essentially, can you think of the cell as a sink to the substrate? And so the biology brings in the substrate from the outside to consume it, creating this gradient. So in numerical space, what this means, so for those of you who are not familiar with numerical models, essentially it's discrete points in space. So in this particular example, I have 9 points that define the area from the inside of the center of the cell to the outer cell, and for each point I can calculate this diffusion of the substrate of interest. So in this case, let's say it's CO2. And so, oh, there's some stuff missing there. So some equations have disappeared there, I'm sorry, but the equation 1, what it is that delta G. It's this chemical voltage that we know how to calculate, and equation 2 is the rate. So you can calculate the rate of consumption of CO2 by making some assumptions about the hungriness, should I say, of the cell. So this in terms of metabolisms and thinking about enzymes is that the Vmax and Km that you find in enzyme kinetics. So applying this Michaelis-Menten enzymatic kinetics to metabolism is something called monokinetics. And the equation should be there. I'm sorry that it's not. And you can relate that to the concentration of CO2. So if you multiply these two together at each of these individual points, you get a power, right? You get a current and a voltage, and you multiply the two together, you get a power. And then if you have a 1D model, you would add up all the powers at the different grid points. But since we're interested in a 3D cell, you integrate over the volume of the sphere. And that gives you the power of the cell. Or you can think of it as the energy consumed by the production of methane, which is the same thing as, the energy consumed by the production of CO2, which is the same thing as the energy released by the energy consumed. Anyways, you get the idea. It's the power is the important parameter here. And so you can add a column to the table that I showed earlier. I'm just using the first 3 springs. And so on the top right, you have again that delta G, that voltage. And then on the table on the lower half, you can calculate the power generated from this modeling approach, and you can on the column after that you have that power demand, which is that maintenance, which is simply an Arrhenius relationship that is a function of temperature. That relationship is lab based, and so it's best we can do for now, but it's definitely a big gap in knowledge and how to really understand what that minimum maintenance is for a particular cell in the natural environment. And so if you take the ratio, you can see here that it's quite interesting because it gives you an assessment of habitability that did not exist when considering only the delta G. So if you look at LCHFB, you'll see a delta G of -55, which in principle should be good for biology from a voltage perspective. But when you include the power, you see that the habitability index is in fact less than 1 at 0.007. And so what this means is that in fact for metabolism of methanogenesis that uses CO2, then it's actually not happy there at all. And the reason is because if you look at the temperature of that LCHFB, you see that it's at 91 degrees. So the maintenance energy is quite high at this point. So the energy you receive from the environment does not compensate for the energy that you need to just stay alive. So that's cool. So that's one way to use this kind of power concept is to add a column essentially to spring geochemical data. Another way is to kind of link geochemical models to predict hydrogen given a water-to-rock reaction using software like EQ36. I started off using Geochemist Workbench, GWB, but it actually doesn't include iron brucites, nor does it do solid solutions. So it's actually it's not ideal to study serpentinization reactions. So this is why EQ36 is better for that. And so you can link EQ36 with these models of bioenergy and kind of explore the physicochemical landscape of this habitability. So one has to be careful when doing that, because the membrane is actually very important in how it transports different substrates into the cell. So if you look at the equation on the left, the metabolic equation on the left, so CO2 plus 4H2, those are kind of neutral species that kind of diffuse through the membrane, right? So that means the entire surface area of the cell is open to the transport of hydrogen and CO2 because they're very small molecules, just diffuse through. But if you look at methanogenesis that uses bicarbonates, then bicarbonate then cannot just diffuse through the cell. It goes into the cell using ion channels, which are called porins. And so you have to take that into account when quantifying the diffusion of the substrate into a cell. So this is what a porin kind of looks like. It's a protein which has like channels inside it that allows transport of these ions into the cell. Billy, I hope you're proud of me that I have a presentation where I cite the Brock biology of microorganisms. And so you can use these parameterizations to kind of change, I guess, the diffusivity coefficient of the membrane. So in this case, because only 0.75% of the cell surface is actually open to the transport of these ion channels. The way I address this numerically is to just dial down the diffusivity of the membrane. And so if you go back to this picture, one also has to be mindful that what you measure in the field is not CO2 or bicarbonate. What you measure in the field is DIC. And so you have to make sure that you speciate the DIC properly, because as you know, the speciation of DIC is pH dependent. So DIC will preferentially go into CO2 in acidic environments, will preferentially be in H3O3 minus in more neutral environments, and then at alkaline environments, and particularly in hyperalkaline environments, there are essentially no CO2 to speak of. So you kind of can already have an intuition here that at high alkaline environments, the methanogenesis metabolisms that uses CO2 will not be operating very well. Thank you. Yes, I'm almost done. And so if you plot this, so these are kind of the results of this numerical model here. I have the pH on the x-axis and the hydrogen on the y-axis, and you can compute the habitability index for these different metabolisms. So you can see that on the figure on the left, you have essentially the environment that has high pH is not habitable for the metabolism that uses CO2 because of the speciation factor. But if you're using bicarbonate, then you should be fine. So you can merge those two together, if you will, in creating kind of energy-derived metabolic maps, I guess, where depending on what your geochemistry is, and if the biologists using 454, 16S, or other combinations of letters and numbers to identify what metabolisms exist, then you can plot there, you can anticipate what particular metabolism the cell is using. So this kind of map is a cool way to link the geochemistry and the microbiology together. And so I have a few more minutes. And so you can also play around with temperature. So what I do is just changing the right plot, where I increase the temperature from 20 to 50 degrees. This time is the same metabolisms. And of course, at higher temperature, your maintenance energy goes up. Your habitable space goes down. So since everybody except Freda included the Gretchen's picture of serpentinite, I thought I'd do it too. If you're interested in this, in methanogenesis in particular, this is the kind of the data I need from you in the field. If you're interested in other metabolisms, please include me in your proposals. And to finish off then, this method of including power provides a higher resolution assessment of habitability compared to the delta G alone, although just calculating delta G is quick and gives you a broad view of the landscape. And then you can target in a particular area using this method. And finally, I think it's a cool way to put the microbiology efforts within the geochemical context through these energy-derived microbial maps. So that's it.
Thank you very much, Sanjoy. I'm looking forward to reading the paper when you get this published, so I can.
Yeah, Polaroidal
I just had to
2025.
get a dig in there. So we have one question here on the web. Let's answer that question. From Frieder Klein, he had when you did the calculations for Lost City, did you use the in situ pH to compute the energy power landscape? He says the ones you measured are the listed are the measured ones. I think he may refer to the in situ down subsurface. In situ, which...
I don't remember what that value was taken from. I think it's just at the vent, but I'm not sure. It's a good question. Maybe others
okay.
know work of Lost City?
Quickly then, there was one other question. What about acetate fermentation to estimate habitability? You considered only CO2 reduction.
I haven't looked at acetate, but I think it's a logical next step to investigate. But how does acetate go through the membrane is difficult. So one of the challenging aspect of this type of approach is you need to understand the biophysics of how the substrate of interest goes through the membrane. And so, which is why it's not trivial to get these data. So anybody who has some expertise in the biophysics of membrane transport, I'd be very interested in working with you.
Okay, I think if there's any further questions, it's good to interact with Sanjoy on the web chat there. I think to keep things on, we'll now move to Mike Russell, to the next presenter from JPL. And Mike, if you can zone back in.
Yeah, Mike, actually, if you could start with your webcam first. I'm going to stop your screen share. If you could actually start your webcam first, then we'll do the screen share.
I thought I'd done that, sorry.
There we go. Okay, now go ahead.
We can hear you fine. Yeah, there you are.
And right, now I do this. Share my screen.
I'm just going to ask, I know how passionate you get about things when you start speaking, but just please try to keep things on time today.
Well, I am normally not too long anyway. Anyway, right, can you see me?
Anyway.
Can you see it? Yes?
Take it away, please, Mike.
Okay, so I used to think, and I think a lot of people thought, that maybe serpentinization might have been a kind of proto-life. And what I'm going to try and talk about in this short talk is that it's absolutely not proto-life. That serpentinization, I think, is the mother of life, but it's definitely not its sister. So now what do I do next? How do I turn the next, how do I get the next one, roughly?
You can either click or use your keyboard.
Okay, I got it. Okay, so I think everybody's seen a form of this structure of how plate tectonics or perhaps ocean floor spreading or continental drift might work. But the point about it is that it is an engine. Whatever you think was happening around about 4.3 billion years ago, it would have been making new ocean floor of some kind, even if it was just with plumes. And that would be, and we should think of this as an engine. So this is the primary engine for us to worry about. And of course, the engine gives us positive feedback because of the high density of electrolyte, which gives you the positive feedback of giving you the body force that brings the material down as the basalt magma or whatever magma it is rises because of its relative buoyancy. But you need both for the continuation. So if we have that sense, then of course, we can now switch to saying, right, what's a serpentinization engine like then? And the serpentinization engine is basically, and I'm sure much of this has been covered, is it's suffering. It's partly due to the convective stress that it's suffering due to the plate tectonics or whatever, or plume tectonics, and so that cracks the rock cracks, the water comes down, it increases the permeability, it increases the pore pressure, so you've got a positive feedback almost immediately. And of course, as we know, it's exothermic. It reduces water to hydrogen, as we've heard, and maybe slowly reduces CO2 or bicarbonate to methane. Perhaps that's slow, but anyway, the serpentinization engine gives us hydrogen, some methane, and some formate. So that's, to my mind, the mother engine of life. So we thought we'd have a look at this kind of serpentinization reaction, but within a disequilibrium experiment. That is, that we're looking at not only, so we're not doing a single pod experiment. It's a two-pod experiment. We switch every 4 hours. We have it about 120 degrees centigrade. And the one extraordinary thing to us, anyway, to my disappointment initially, was that we'd made no methane using CO2-13, so carbon-13. And of course, at the same time, pre-publishing us is McCollum and Donaldson's paper, which comes to the same conclusion, that we don't make methane, at least quickly, in serpentinization reactions. What we do make is formate. So you could say that the formate reaction tells us something about CO2 reduction to formate or CO, and that could be perhaps segued into one part of the acetyl coenzyme A synthase pathway. So here's the fact of this, and that is that we've got, and we think this is fair enough, we've got hydrogen, the hydrothermal solution interacting with the ocean solution every 4 hours switching, and you can see every time the hydrothermal solution comes in, we drop methane, but every time the ocean comes up, the ocean solution is put into the serpentinization reaction, then we get not only the magnesium coming through, but also the formate. So it looks as though that's a very rapid, I won't say instantaneous, but extremely rapid reaction. So I don't want to go through this particularly, but just to remind us that we should think about the serpentinization as an engine. It's an autocatalytic collective. And I'll just give you a few of these, which you've probably had. The mafic crust becomes unstable because its resistance to tectonic stress, because it fails and becomes unstable due to the tectonic stress, and the cracks form and compete and multiply and so forth. And one of the things that I think is open to, I think we've done the rest, but negative feedback, significant, what we would like to call the governor or thermosetting. And I think there's some disagreement about that, and I've heard some of the disagreements so far, but I thought the Prostierowski paper suggesting 140 degrees centigrade maximum of the hydrogen isotopes makes a lot of sense to me, and the carbon in the carbonate perhaps makes less so. So we would think that that's due to softening of the crust, and that's why we get a thermostatic system for these lower temperature alkaline non-magmatic systems of around about 100 degrees centigrade once they come towards the surface. And I've just emphasized that these systems last for about 100,000 years, and they last, I prefer to call it 10 to the 21 nanoseconds. We have to think on the pico and nanoscale if we're thinking about the emergence of systems such as life. We partly deal with this in a paper just coming almost out, should be out next week, Branscombe et al., escapement mechanisms and the conversion of disequilibria. So our story is all about disequilibria and the engines of creation. So this is what we used to say, and I feel embarrassed about it, but we suggested that perhaps methanogenesis was the beginning of life, and therefore serpentinization was significant to that effect. So here we have it on the right-hand side. Perhaps CH4 was the first formation of the first autocatalytic metabolism system, but we no longer think that's the case, partly because we can't make it very quickly. And anything that is chemical that we're going to try to bring into use for the emergence of life must be fast, almost instantaneous, but this is not true of methane. So we've kind of given that out and now suggest that we go back to looking at the early Earth. This is not a very good diagram, but what it shows is the hydrogen, methane, and hydroxyls of an alkaline solution are well out of equilibrium with the acidulous ocean, but also with nitric oxide, which we would say is sufficient to produce nitrate in the ocean, something to about 10 micromoles, which is enough to, we would say, use up, actually burn, if you like, the methane and the hydrogen. And so in other words, we are going to suggest methanotrophy first, not methanogenesis. Unfortunately, methanogenesis has given, because it had prime of place, we think of methanotrophy as reverse methanogenesis. But I would ask you to rethink that and say, well, could it be that actually methanogenesis is reverse methanotrophy? Just a thought, but one to bear in mind, I think. So if we move on to what we look at in the actual membranes that are produced, now, as you know, much of the precipitates at Lost City are basically, well, the first precipitate is brucite and some carbonate, but this would not have been the same in the early ocean, where, of course, there's enough iron in the early ocean to make a brucite-type mineral, but in this case, it would be a green rust. And the difference between that is it's a double-layer hydroxide, which uses the anions to get charge equivalence. So if we just take one known example of using this kind of brucite-type mineral, green rust, and we suggest that we put it as part of the membrane, then on the outside we've got nitrate, carbon dioxide, low pH. On the other side we've got high pH, and we've got hydrogen and methane. And the methane, we won't go into that right now, but the two are obviously very far from equilibrium. And we would suggest that the nitrate, what the nitrate does is it oxidizes the green rust. And of course, as soon as it does that, it opens up a kind of craw for the nitrate to get in to balance charge. And if there's more nitrate on the outside, then of course it'll be forced in and be reduced to nitrite as the brucite gets oxidized, and eventually it'll go down to HNO, which is uncharged, and therefore you can kind of push it through because it's loose. It'll be loose within that interlayer, and the protons will help to drive it through and help to reduce nitrate to ammonia, and that can be done in 3 hours just by bucket chemistry. And nitrite can be reduced to ammonium in 1 hour, and I think that that's hugely significant because we always think it's just CO2, and of course I do like to say that life hydrogenates carbon dioxide, but it also makes an aminating compound as well from nitrate, we would say, and that's a very good source of nitrogen for amino acids and nucleic acids, which is otherwise missing, by the way, for most hypotheses. So we think of that as a ramjet. I won't go into that. But so the real problem facing us is how do we get from a hydrothermal mound to the last universal common ancestor, and then eventually the evolutionary tree? How can we? And I've kind of made a figurative kind of a canyon here to try and cross. How can we, on Earth, can we get from the left hand to the right hand of this diagram? There's lots of things going for it in terms of pH gradients and so forth, and of course the gregite being formed very similar to acetyl coenzyme synthase and carbon monoxide dehydrogenase. But really, I think we've been missing the point. It's going to be hard to get right across, and I think we need a vehicle. And what do we actually need to get to? Our aim must be the ribosome. We've actually got to get a ribosome. We can't mess around with a little bit of metabolism and then try and look for information system and so forth. We've got to have everything together, I would say, and therefore we're aiming for a ribosome. Now that's a big aim, so let's see what possibility we have here. And what we'd say was the first ribosome, according to Bernhard and Tate, could think of entirely in terms of peptide. And in fact, the ribosome has an ancestral ribosomal peptide anyway, with plenty of glycine in, and that's a linear peptide. And it looks as though, according to, shall we say, Williams et al., Loren Williams, that probably it could be that the peptide was first and that the RNA and DNA came later. And I would certainly agree with that. In fact, I don't think RNA was significant at all. I think it went probably straight to the nucleic acids. Anyway, so what can we do here? Well, we've got hanging in the on the curtain, so to speak, is green rust. So can we, and green rust is that double-layered hydroxide, which can take in the anions, as I said. Now, can we get across this canyon using green rust? Well, let's have a look at something that was suggested, by the way, in 1903, that in fact, Darwin's system, Darwin's evolution absolutely had to have a governor, had to have a guidance system. And this Berkeley in 1903 suggested, he was just a laird in Scotland, in some estate in northern Scotland, and he suggested, well, how about comparing it to a Jacquard loom? And that was invented in 1801, and of course that's what inspired Babbage and Ada Lovelace to go for that digital idea of the difference engine. But actually, that was 40 years later. But the Jacquard loom is not dissimilar, as actually Drexler pointed out some time ago, that because of what it does is it automatically makes material. The output is material by this automatic machine. And it's extremely, well, I shouldn't say extremely, but it's comparable to the ribosome and how the ribosome works. So on the right-hand side, you've got those digital system going through, and it generates, the output is protein, newly born protein, or peptide anyway. And on the other side, there is the material. So if we then look at green rust and we look at Jeanine and Ruby and other people's work, you'll see that actually it's extraordinarily complicated system which can vary. So what we would say is that ferric iron could be thought of as the ones, and the ferrous iron could be thought of the zeros. So you've got a 0-1 system in green rust as it oxidizes and reduces. Now, of course, it's a dynamic system, and it's an oscillating system as well. So it would start rather complexly. It's not going to start just by additions. But I think that probably is a good thing rather than a bad thing. So here's another example of one of the readouts you get from green rust, and there's a comparison with the Jacquard loom inputs, basically the delta ones. And so we then appeal to Greenwell and Coveney, a lovely paper in 2006 in the Origins of Life, where they show that in fact you could use green rust as an information and storage and transfer system. And if we put that across the membrane, there we've got this kind of system which could give you a storage and information. They, of course, employ DNA and RNA, and we would suggest that wouldn't be the case. We would suggest that this would actually control through diffusion and so forth, and reaction rates. It would control the generation of particular organic molecules, and those that are the most successful and interact are the ones that will continue, and those that don't interact are just pushed to the side, perhaps as amyloid or peptide. So here's a system in a little more complex way. So the nitrate goes in on the left from the ocean. It's reduced by the protons and the electrons from the alkaline hydrothermal system through this semiconducting mineral, green rust. And you can see you can get down to ammonium. The ammonium then can get to react with the I haven't quite got this here. So I've got the reaction. We get the methane coming up, and the methane can react with the nitrate, and the nitrate perhaps could reduce the methane to a methyl group. So then you've got a CO being generated in the same system and a methyl group being generated in the same system. So here it is in its relative entirety. Nitrate to ammonium on the top, through the top layer, and then the next layer is the nitric oxide. Halfway along that top layer is used to oxidize the methane in a very complex way that actually employs electron bifurcation to give you the methyl group and the aminogen, and the aminogen can react with the something like pyruvate, for example. George Cody's given us pyruvate, and Laurie Barge has used pyruvate in her reactions to show that you can actually make alanine almost immediately. You can make polyalanine if you would use a carbonate, and so you've got a very short peptide, something like 4 or 5 or 6 mer peptide, which is enough to do an awful lot of sequestration of phosphates and sulfides and so forth, and even enough to make a kind of membrane around the outside. Now, this is hurried, but just to give you where we're going with this is to suggest that actually we've got a serpentinization engine that spawns life's engine, and that serpentinization gives us hydrogen. crustal leachate gives us the methane. The two together, interreacting with nitrate through proton motive force and electron bifurcation, gives us carbon monoxide on the one side and methyl group on the other, and you get to the target molecule for example, the Krebs cycle, the reverse Krebs cycle, the incomplete reverse Krebs cycle, gives you the acetal group, and we would say that's, once you've got there, you're off. So we would say maybe that's the way to do it. We actually try and do it in a one-er. We don't kind of go through using little vesicles or anything like that. We actually say those membrane minerals with their gated mechanisms actually do the real job of the cell. And so we would take the extreme view that if you want to look for the original cell, it wasn't unlike a green rust, which are all about the same size, by the way, depending on the interactions, but generally at the micron or submicron level. And these are the people that help me a lot with all this, and Patrick Beckett at the top, Wolfgang Nitschke, and Albert Branscombe, who I believe or hope is online and will be emailing me thereafter. Please, Albert. Okay, that's it. Thank you.
Wow. Thank you very much, Mike. I think I see everybody's hair appropriately swept back after that, that tour de force presentation. And we did have a couple of questions that came up during the talk. Maybe we can address those. Frieder Klein asked some questions about the green rust. He asked, would you mind explaining which reactions led to the formation of green rust in the Hadean Ocean, and what kinds of green rust would you expect to form?
Well, of course, that depends on which whereabouts you are in the membrane and how much interaction you've done with the oxidants, such as nitrate and nitrite. But what we say is that, of course, I believe even the Pony deposit in South Caledonia has brucite being precipitated to this day at that alkaline vent. And what we would say is there would be enough iron in the early ocean to give you brucite at the precipitate, where the acidic ocean would meet the alkaline vent. And as an excuse for saying that, or a suggestion for saying that, Steve Moyges and Gustav Arrhenius pointed out a long time ago that probably all the banded iron formations of the Archean and the early Proterozoic were all originally green rust, that was the major precipitate anyway in the early ocean. And we would say that would be very particular at an alkaline vent, of course, where it would be irresistibly dumped in place of, after all, brucite. So brucite today, in those days, it would be a brucite-like molecule, but it would be probably ferrous to ferric high ratio. But as soon as the nitrate comes to bear, then it can go to ferric. And in fact, so long as it's well protected by phosphate, it can go all the way to ferric and yet get reduced again. So it's just like an enzyme. It can go from almost entirely 2+ to almost entirely 3+ and back again.
Okay. So Eva Stoikin asked, could you use sulfate in this scenario instead of nitrate?
You can use sulfate, but sulfate is not reduced in this system. The redox contrast isn't great enough. And anyway, sulfate reduction is not found in the last universal common ancestor. It seems to have been a discovery of the Archaea, and there was then gene transfer. So we rather eschewed sulfate, although we would recognize that sulfate would make the ocean, as Scragg says from Harvard, even more acidic.
Okay, one more question is kind of following up on that. Eva asks, how do you avoid loss of nitrate by reduction of N2? Reduction to N2.
Oh, well, of course, there's going to be different class reactions, but the actual reaction that mostly takes place in the green rust is from nitrate and nitrite through to either some nitric oxide and ammonium.
Okay.
Nitrogen doesn't get a look in.
Okay. Thanks very much again, Mike. And if anybody has any further questions, you can follow up with on the chat room. But we're going to move on now to the pop-up talks. I think we have 3 lined up. And for the first one, we are going to zoom across the ocean to another continent, back over to Europe, and get some talk talk from Benjamin Boutel. Are you there, Benjamin? We have your slides.
And Sam, if you want to double check that we didn't mute him before, in case he was one of the noisy mics. Benjamin, if you can hear us, please maybe use the chat to let us know you're online. If not, we can come back to you once you get set up.
Yeah, my phone lines are all open and clear.
Okay. All right, we're going to, oh, there he is. Okay. We have a couple extra minutes. Want to wait for Benjamin, or should we move?
Yeah, we can do wait just a moment for Benjamin. And then what do we have after this? We have Wes Swingley and Slava, whose last name I'm not even going to try to pronounce what it might be.
Okay, and then Benjamin, in the meantime, too, you can start your webcam. There should be a start webcam button above Tom's head.
Kind of. No, other hand. Other order. Right. This region above Lisa's head.
Okay.
In actuality.
He found it.
Ah, there he is. Oh, he's
okay,
online.
you can see me?
Yes.
Yes. And then
we're all set.
And then
Benjamin, you can hear me?
Slides,
okay.
Yes, we can hear you. Click the down arrow icon below to the bottom left of your slides. That's the best way to advance. Thanks.
Okay, like this. Okay. Okay, so the idea is to explain to you how we see the serpentinization on Mars, and to present to you how we try to work on serpentinization on Mars. So I will skip those slides to show you directly some detection of serpentine and to show you the context of the detection. So here's an example of one crater where we detect serpentine, and it is found with in a station with a lot of over iron magnesium rich phyllosilicates, what you can see here in red, and some carbonates. And the different phyllosilicates that we can find mostly there are saponites or other kinds of iron magnesium snaketites, and sometimes talc. And so the typical assemblages where we see serpentine is a station with iron magnesium rich phyllosilicates and carbonates. And so if we take a look at a global map of detection of serpentine, where it's where you can see this green symbol on this map, we can see that the serpentine are always detected with carbonates, which are the purple symbol in this map. So this is something that is found every time we found serpentine, we found those carbonates together. So the idea is to try to understand how we can make this serpentine and the carbonates together on Mars. And so even if we can also try to understand what is with this process, if we can store a lot of water or CO2 in the crust, or if we can produce a lot of methane and hydrogen, which is also really interesting. I think the first step about serpentinization on Mars is to try to understand the conditions that are needed to reproduce the same mineralogical association that we see on Mars. And this is work that we're trying to do here at SEED. So the two methods that we employ to do that is on one way to do some geochemical modeling with AQ36. So this is here one of the examples of a result that we can have. So this is an interaction between a fluid of pure water and a rock rich in olivine. So it's basically basalt, but really rich in olivine, 30% of olivine, 30% of pyroxene, and 40% of plagioclase. We make the interaction between this rock and a pure water fluid at 200 degrees C at a water to rock ratio of 10, and we add also 1 bar of CO2 to make the carbonates, and we see that the serpentinization occurs only at low temperature, and we can reproduce this kind of mineralogical association that we see on Mars, the chlorite, sometimes the talc, the serpentinite, and the carbonate together with the serpentine. So this is our first attempt to try to reproduce this mineralogical association that we see on Mars. And the idea now for the following work that we will do is to try to do the same work but with experimental studies. And the idea is to work in a closed system with some olivines and sometimes mixed with pyroxene and glass together, and to heat the system at 150 degrees C, and to change the atmosphere that is used sometimes without CO2 or 10% of CO2 or 100% of CO2 at 1 bar, and to try to see in which system we can form the typical mineralogical association that we see on Mars. So unfortunately, we still haven't started this work of experimental work, but hopefully we will start that the next week, and maybe in few months I will have a result to present about that. So yeah, that's it for me.
Thanks a bunch, Benjamin. Did anybody out there have a question? Adrian Brown is interested in how much talc gets formed in your simulations there.
Okay, so here you can see it's a percent of mole by percentage of mole of minerals. So it's around 20% of talc that can be formed here.
All right, great. Thanks very much again, Ben. And we will now move on to the next pop-up, which is from Wes Swingley.
And Wes, if you can hear me, you can go ahead and start your webcam and then unmute your phone's mic.
Yeah, I'm unmuted, and I tried to start my webcam, but it does not seem to have worked.
Oh, sorry, I didn't actually make you a presenter just yet.
I was. It
okay.
just kicked off, I think.
Now please go ahead. There
All right.
we go. All right, take it away.
All right. I thought I would give a talk after hearing Matt Schrenk mention one of my study sites yesterday, and the thought, of course, is to give a talk in a serpentinization workshop that has nothing to do with serpentinization. So this first slide is showing our study site at the Calumet Wetlands, which is on the south side of Chicago. And we call it the surface of the moon, and you can kind of see why. And this is a dumping site from the Industrial Revolution here in Chicago, which is a leftover remnant of a lot of infilling that the city was doing intentionally to try to reclaim land in this historically wetland area. And so, as you might know or not know, Chicago is off of Lake Michigan, and this study site is a literal rock's throw from Indiana in the south side of the city. And the aerial view here is showing, the Google Maps view is showing the extent of this slag site, which is quite large, and as far as studies have shown, meters deep, several tens of meters deep, that was filled in all the way up until the mid-'80s when industrial waste dumping was not as cool as it was anymore. And you'll recognize probably some of the chemistry that is going on at the site. And much like Billy Brazelton has said a few times, I am a microbiologist, and the geochemistry is done by other people. But the geochemistry here is relatively similar to the serpentinizing systems, where you have calcium or magnesium silicates that are being weathered by water, in this case meteoric water and ultimately groundwater, to yield a high pH system. But the chemistry is very different in the products coming out, and that is we have high calcium fluid, relatively freshwater fluid that has high hydroxyl ions that are leading to the high pH. And the table on the bottom left is showing some of our study sites that show the extent of this pH at Site 2 and Site 6, for example, are groundwater wells, where our pH is up above 13, and we've seen it reach as high as 13.4. And the systems are, as a nature of being a steel waste site, are loaded with a lot of metals as well. Lead primarily is fairly high, and magnesium and calcium are both off the charts, as you might expect from the chemistry. And just taking a look at the site, anybody who's been to one of the terrestrial alkaline sites would recognize a lot of the same features here, where we have a calcite precipitation throughout, and this is a irrigation canal going through the city, so as such it has very steep sides. One of my students here from an RU project that I did is excitedly testing the depth of this just off the shore, and you can't quite see it, but that grabber stick went about 5 feet deep in the sediment just a couple feet offshore. So it's very deep, and the calcite precipitation is very reducing in that we have completely anoxic conditions just millimeters below the surface. We've done a lot of analysis here and unfortunately haven't gotten anything published yet, but a couple of our primary looks are at the transects along this system going from in the shore into this calcite precipitation, and also on the vertical core going down through the deepest layers of the anoxic sediments. And without going too in depth on this, what we see is very similar to what Billy and Matt have described, and Darcy to some extent as well, have described in similar natural systems, where we have large concentrations of beta proteobacteria, primarily relatives of the Serpentinimonas and Hydrogenophaga that was mentioned before, in the aerobic sediments, and that gradient transitioning to anoxic system that is dominated by firmicutes, and that's the bright red that you see on the right hand of that chart, where we have heavy firmicute populations in the deepest layers, and we're looking at metagenomics and 16S ribosomal surveys to see exactly what organisms are there, and in the case of some of these, doing assemblies from the metagenomes in order to analyze more of how they're surviving in this high pH. As Matt said, this is an open question. Nobody really knows why or how these organisms can survive in these systems. And then the question of whether they're thriving or merely getting by. And that's basically it. So I'm happy to take questions. I see Darcy's got a question.
Yeah, Darcy sent in the question, okay, what happens at depths between 25 and 30? Is that the aerobic anaerobic interface? So it looks like there's quite a change in the microbial populations.
Yeah, for sure. And the line that I've drawn there is the rough shoreline, and the shoreline, of course, changes with the water level as well. And so that level really does seem to be the transition where you go from a slimy biofilm on the shore into a shallow depth biofilm that then changes into the calcite precipitation. So that 25 to 30 change there is where we see the calcite precipitating, and everything above that level is basically too disrupted by the flow of this river, so that there's a constant few centimeters of depth of water over the top of this calcium carbonate precipitate. And that one, to answer her other question, that one's a horizontal core, or a horizontal transect rather, taking 5 centimeter increments. And we do have a core that looks very similar to this, but the top layers of the core, when we get out into the water, don't actually look a lot like the shoreline, where we don't have a huge concentration of these beta proteobacteria in the upper water column like we do on the shore. It seems to be a surface associated.
And Billy just commented there that it's interesting to see that we have this very similar microbial populations to what we're observing in the serpentinite system. So
yeah, the geochemistry is similar in some ways, in the pH, of course, and some of the anions and cations, but there are some major differences, and we're really interested to see what this means in terms of biogeography and also in terms of survivability for the high pH systems.
Do you know if the slag there, is it driving any kind of like hydrogen production or?
We don't know, and yeah, and this is a limitation of my own crappy geochemistry, that we've not done some of that core geochemistry on methane and hydrogen. In a lot of the summer seasons, we do see bubbling coming up through these precipitates, and we've tried to do some studies on methane production, but we have a lot of sulfides in the environment, and the sulfides were cocking up some of our machinery.
Also, I was curious to know, like, what was the highest pH that you've gotten, like, DNA out of it?
We get DNA out of everything, and the highest pH that we've measured is both in the groundwater wells and in some of the springs. So there are just little groundwater springs that leak out into the slag. The big slag pile is basically behind the student that you see in the image here. And the springs that come out of those, we've reached as high as 13.4.
All right, great. Thank you very much, Wes. We'll go to
a
our
lot.
last pop-up now, and we're going to Slava, and I will let him introduce himself.
Hello, everybody. Can you hear me?
Yes, we can.
I'm looking where to start. Okay, I see webcam here. Start. Okay, can you see me?
Yep, we can see and hear you. Just take it away. You can control your slides by clicking the down arrow icon located to the bottom left of your slides there.
Okay, I see. Great. So good evening from Paris. My name is Vyacheslav Zgonik, and thank you for this opportunity to present our work, and thank you also for organization of this workshop in form of web meeting, so we can participate overseas. So I'll present our findings on sea pages of natural hydrogen. Here's one picture of Atlantic coast of the USA, and there are a lot of interesting formations on its surface, so-called Carolina bays. There are a lot of them, about 500 of thousands of these structures. These are oval depressions, shallow depressions on the surface, and they range in size from 100 meters to several kilometers. And we tested them for presence of hydrogen gas, and we discovered that in subsoil layers inside of those bays contain molecular hydrogen, while outside there is no hydrogen at all. And we studied in details this discovery, and we published our results recently in a peer-reviewed journal. So I will show you several examples now. Here is one average sized bay, and the red line shows the profile on which we measured hydrogen in subsoil gas, and the green graph, or green line, shows hydrogen concentrations. And you see once we enter inside the bay, they increase many orders of magnitude, and once we leave, once we go outside of the bay, there is no hydrogen at all. And this is the same place showed on LIDAR image, showing elevation in artificial colors. And here's another place where we tested for hydrogen. The first one was in North American continent, this one in Europe, in European part of Russia, just near Ukrainian border, where I'm originally from Ukraine. And here we studied one large depression on the surface. The red line represents the profile of measures, and on the right side you can see, not sure how to make the arrow, but on the right graph you can see the hydrogen concentration, which corresponds to the red profile line. And once we enter into this depression, concentration increases abruptly. And on the lower left part, you see the elevation profile, which corresponds also to hydrogen concentration. Of course, we verified that this hydrogen is not superficial, because it was first assumption. We made a lot of tests to see where it comes from, and we have evidence that this hydrogen is of deep origin because we have results from wells drilled nearby of such depressions, which are very hydrogen rich. There are tens of percent, sometimes more than it's a major gas. Also, recently we tested for other fluids, tracers like helium or mercury, and we also find that their distribution corresponds to hydrogen, so their concentrations are very high inside of these places, these locations. And based on data we obtained from field tests, we estimated daily flows from these places, and they're actually very high in some locations. It's our numbers give up to 21,000 cubic meters per day, which is very high. It's comparable to production from natural gas wells. And we published those results too. And I will show you one more example. Here is the very small structure, only 100 meters in diameter, and there are 3 profiles crossing it. And you see the same distribution. Once we're inside, we have this very high hydrogen content. So we believe that these locations are some kind of chimneys used by fluids, or like a pathway for fluids. Probably it has a relationship with hydrogen because hydrogen is a reactive gas. It may have reacted with minerals, creating porosity, and the porosity collapsed with time. And that's why we see depressions in the same time, hydrogen concentrations and other gases as well. And so we tested these, many locations. Unfortunately, I don't have enough time to present everything we've done. So, but our collaborators tested for hydrogen in South America. So we have 3 continents now where these locations are present, and the numbers of hydrogen are very, very impressive actually. And we decided why not to. We wanted to know more, and to know more, we need to drill in those places. So we created a company, Natural Hydrogen Energy, where I'm working on now, and we are planning to make deep exploratory drilling into one of these locations we selected in the United States. And this very good time because just 3 years ago in Mali, in Africa, there was a proof of concept. One drilled well started to produce 98% pure hydrogen, and they used this hydrogen to make electricity to supply for a local village. And so we want to do something of the same nature, but in the United States. And therefore, we're working on this project now, and because our team are mainly scientists, we're all researchers, and we want to do a lot of research connected with this project, and that's why we're looking for collaborators. So, for example, we want to know what microorganisms are living associated with these places. Maybe it will be possible to start from surface tests, and then when we will have deep samples to study what's going on inside. Or, for example, there is a need to study reactivity between rocks and hydrogen, because there are a lot of research ongoing, what we've seen today on production of hydrogen from minerals, but not enough actually of how hydrogen reacts with other minerals. So I invite you to contact me if you have any interest, and please check our references. And if you have any questions, I will be happy to answer. Thank you once again for this opportunity to be present. It's very exciting. Thank you.
Okay, thank you very much. Could you pronounce your first name again?
My first name is Vyacheslav, but everybody calls me Slava. It's more simple. Just please call me Slava.
Yeah, it's easier for us to handle. Thanks a lot. So do you know anything about the geology that underlies any of these sites? Is it?
These are sediments and not very thick. In Atlantic coast, there are about 100 meters of clays and sand. And in the case of Russia, there are 600 meters of sediments, mostly limestones and sandstones.
So
we describe those.
We did have one question here on the
in papers.
Sorry, we did have one question here on the web. In the chat room, it says, it sounds like you're not sure of the source of the hydrogen. Do you think this was different than the source of hydrogen that has been proposed for like deep serpentinization sources? And do you think this would affect the estimates of the global hydrogen production?
Yes, definitely, because there are a lot of these structures around the globe, and this will definitely affect the global estimates for hydrogen production. Is something we're working on now, and it will increase at least by one order of magnitude estimates, and probably even more. But we need to do more research to understand. And concerning the origin, we have two main approaches now. One classical approach is for serpentinization, and one is more new marginal approach that this hydrogen comes from mantle degassing, and this is a primordial hydrogen stored in Earth, inside of the Earth since its formation.
Okay, and one other question, one I also had, I don't recall if you mentioned how you're doing these hydrogen measurements, and can you detect other gases with the same approach?
Yes, we make a shallow hole in the soil using the steel rod. We push the steel rod into the soil, and we take it out, and we plug immediately with the plug connected to portable detectors. So we measure this way in the field, and then we sample in the different types of containers, and we make analysis in a laboratory and gas chromatographs to confirm our field results.
All right, thank you very much, Slava. We do have a couple of minutes left. If there's any other persisting questions out there that anybody had for any of the speakers today, if there's any questions, do I hear anybody else have any other comments or contributions they wanted to touch on? Right, well, sounds like that's it. So why don't we wrap things up for today, and remember, tomorrow we are going to reconvene at the same bat time, same bat channel, and be talking more about things having to do with planetary perspectives on serpentinization. And so see you back here tomorrow, hopefully. Bye.
Thanks, everyone.
Thanks, everyone. And again, a reminder, we're posting the survey link. So we want your feedback. We'll leave this meeting open a little bit longer than we did yesterday. We accidentally shut everyone out. So you can continue posting questions in the chat, and we want your feedback. So you can go to the link there and share your ideas.
Yeah, nice job, all the speakers.